Hello, my name is M.Grande and thank you for coming to listen to my presentation on the understanding of human muscular disrepair and recovery. To start, 1 in every 30,000 people will have some form of muscular dystrophy, which is a fatal disease that slowly and brutally murders its victims. Muscle lesions, on the other hand, although less gruesome, can comprise up to 55% of all injuries in athletes, as well as being slow and expensive to heal. Taking a closer look into the former, muscular dystrophies, such as Duchenne's muscular dystrophy, are genetic impairments that you're born with and are usually fatal before the age of 30. The trickiest part about treating muscular dystrophies is that the dystrophin gene, which is responsible for providing structure to the muscles and is attacked by this genetic impairment, is so large, making it hard for gene therapy to tackle the problem. Moving on to muscle lesions are any sprain or break in the muscle, and they're very easy to sprain and break. Natural healing is slow and it's expensive, not to mention that most of the time it doesn't even heal properly because it leaves behind scar tissue, which is probably the trickiest part about treating it. My first article discusses this issue, and it is titled New Strategy to Facilitate Muscle Regeneration After Injury, brought to you by the University of Barcelona, Spain, with some notable authors being Teresa Pages and Joanne Torella. Some notable takeaways from this article are that hypobaric hypoxia conditions have been shown to have a benefit on muscle regeneration. What are hypobaric hypoxia conditions? Put simply, they're just conditions of low oxygen. Now, this is because the HIF protein in cells, when exposed to low oxygenated conditions, activates many growth factors, such as an endothelial growth factor, which in turn activates other growth factors, which can contribute to muscular healing. This protein also activates mitochondrial efficiency, protein synthesis, and many enzymes, creating a healing concoction for muscles. My second article is titled New Gene Therapy for Muscular Dystrophy Offers Hope, brought to you by the Washington School of Medicine in the United States of America, with a notable author being Dr. Hakim Tassifal. Some notable takeaways from this article are that gene therapy for Duchenne's muscular dystrophy has been shown to be successful. Now, if you remember to what I said earlier, the trickiest part about treating Duchenne's muscular dystrophy is how large the dystrophin gene really is. But the scientists in this study got around that by using 3 harmless virus shuttle vectors, each with 1/3 of the dystrophin gene, so that they could be implanted in the cells and reassembled inside. And improvements are still being undergone, such as lowering the number of shuttles needed so that immune responses are a reduced risk, but results are promising. My third and final article is titled Promising New Gene Therapy Approach for Genetic Heart Disease, Clinical Trials Imminent, brought to you by the Hubrecht Institute in the Netherlands, with a notable author being the one seen on screen that I'm not going to try and pronounce. Some notable takeaways from this article are that new gene therapy for the genetic disease ACM has successfully restored protein levels and rhythmic contractions, which gives you a clue to what ACM really is. ACM is a genetic disease like muscular dystrophy. However, while muscular dystrophy attacks skeletal muscle, ACM attacks cardiac muscle and increases risk of stroke and an irregular heartbeat. Now, while gene therapy for DMD uses the exact gene, ACM is different in that it only uses a similar gene to code for a similar protein, not exact. Next steps in this research include investigating clinical potential, safety concerns, and the efficacy on human patients, because this study has only been shown to be successful in lab environments. After reading these articles, I came up with 3 possible research questions. 1. Would protein injections, gene therapy, stem cell technology, or CRISPR gene editing work best to treat muscular diseases? 2. How could I do a research project about a muscular injury recovery using hypothesized methods while at the same time meeting New York State humanitarian animal laws? And 3, how much of a difference is it to treat skeletal muscle injuries versus cardiac muscle injuries? What about genetic diseases? Some additional resources are pictured on screen. And with that, I can